We had stopped with where we had stopped last time. Right, so we had stopped at with DNA replication, right? So today we're going to be doing two things. Two things I want to cover: transcription and translation. Those online, you can see the board properly. Yeah. Okay, and this following something we call the central dogma of molecular biology. Right? And this states that DNA is used to produce RNA, which is then used to produce proteins. Right? So everything in molecular biology is based on this. You have DNA is something is born with. Certain parts of your DNA will be converted to RNA, which are then used to produce proteins, which give you your physical characteristics. Okay? And it have two processes in which we perform this. The first one is transcription, and the second one is translation. Right? So the first one we're going to start with is transcription. I've given you all, what is it called? So the end of last class, I would have given you all a little summary of how transcription and translation would be, right? That was this diagram. You all have that diagram, right? We DNA inside, transcription to go outside, and then translation. Here, today we had to go in a little bit more detail. We have to know the enzymes involved and the exact mechanism. I've given you all this last class to the central dogma. Okay, if you all know how to just take it down, right? And then we'll start with transcription. Just take down the central dogma of molecular biology, starting with transcription. Right, so remember your DNA is found inside the nucleus, right? So your DNA is found inside the nucleus and it's found in a double helix, right? Your DNA cannot leave the nucleus as it could become damaged. Once your DNA becomes damaged, you can change the type of proteins being produced. Change the types of proteins being produced, it changes the characteristics of that organism. You don't want that to happen. So, in order to express DNA, you have this mechanism. You get DNA into RNA. The RNA could then be transferred into the cytoplasm, which could then be used to produce the proteins at the ribosome. Okay, so the first thing we have to start with is initiation of the sequence. And to initiate the sequence, we have to use a special enzyme called RNA polymerase. Okay, so what RNA polymerase do, one does, right? Have your DNA, inside the nucleus, right? The double-stranded DNA helix inside the nucleus. Okay, let's say that's the top. What RNA polymerase would do, it come in and will unwind a certain part of this DNA. So the DNA, when RNA polymerase is present, unwinds a little bit. Okay, so this part opens up. Now we are accustomed to DNA replication, both strands being replicated. This is not replication, this is transcription. Okay, so what RNA polymerase is going to do? Okay, let's say this is A, C, T. Okay, that is putting bases there. RNA polymerase is only going to act on one of these chains. Right? What it's going to do is going to come in and it's going to give complementary bases to the DNA. Right? So which base in RNA is complementary to E? U. Remember, RNA doesn't contain any T's. Right? Which one will be complementary to C? G. And which one will be complementary to T? Yeah, complementary to T. It'll be E, right? So you can have A and R. So what RNA would do would go along the DNA strand and make complementary bases to the DNA. Right? This is the transcription part. Eventually, you're going to reach something called a stop codon. A stop codon is a triplet codon that tells the RNA polymerase, okay, you need to stop at this point. At that point, the DNA, well, the complementary bases would leave, the DNA would zip back up, and the process could happen again. Right? So you need a start codon, you need RNA polymerase, and then eventually you need a stop codon. Okay, and what you'll be left with is an immature mRNA strand. Remember, mRNA is for messenger RNA. Yep. Yep. So it's initiation of RNA polymerase, start codon, stop codon, and we have uncyclic. So these are the main processes in initiation of transcription. Okay, so just take a note for it, right? The initiation. Right, so initiation. RNA polymerase binds to specific nucleotides. RNA polymerase binds to specific nucleotides in the DNA. In the DNA. In the DNA, just outside of the gene, just outside of the gene, being transcribed, just outside of the gene, being transcribed. Full stop. This nucleotide sequence is usually TAC. This nucleotide sequence is usually TAC, which signals the start of transcription. Let's stop. The DNA then unwinds. The DNA then unwinds. The DNA then unwinds, comma, exposing the two strands of DNA. Exposing the two strands of DNA. Full stop. One of these strands serves as a template. One of these strands serves as a template for the synthesis of mRNA. One of these strands serves as a template for the synthesis of mRNA, full stop. This strand runs in a 3' to 5' direction. This strand runs in a 3' to 5' direction. This strand runs in a 3' to 5' direction, while RNA polymerase moves, while RNA polymerase moves in a 5' to 3' direction, while RNA polymerase moves in a 5' to 3' direction. Full stop. Okay, so that's the initiation. So initiation is just starting of it. The elongation is when so this is the initial base pair. It will actually be TAC. So TAC. And so that will be TAAUCG. Right? After you start with this start codon, then the process of elongation can start. Whatever certain codons have afterwards will take part in elongation. So you're making the RNA strand a little bit longer. Okay. Right. So the next one is elongation. So all of this in the handout, right? Just probably not as in detail. Right? So during elongation, other RNA nucleotides are joined together. Other RNA nucleotides are joined together to form a mRNA strand. Are joined together to form a mRNA strand. Full stop. Right, and then termination. Termination. Right, so termination is just when you reach that stop codon, right? So it have about 4 different stop codons, sorry, 3 different stop codons. Once it hits one of them, it tells the RNA, okay, you can stop it. So for termination now, the termination sequence of ATT, comma, ATC, or ACT indicates to RNA polymerase, so it's ATT, ATC, or ACT, indicates to RNA polymerase to detach from the DNA template. To detach from the DNA template. Full stop. The DNA reforms a double helix. The DNA reforms a double helix. And transcription ends. Full stop. Right, so at this point you have immature mRNA. You're still in the nucleus, right? But that immature mRNA needs to leave the nucleus to get into the cytoplasm, where it could be used at the ribosomes to produce proteins. Right? But before we reach there, we have immature mRNA, right? The immature mRNA, before it can leave the nucleus, needs to be modified. Right? And it's modified into mature mRNA. So the difference between the two is the presence of something we call introns. Right? And exons. Right? So let's say the soy RNA is transposed, right? So let's say this is your RNA strand. Right? We're using the shaded one to represent introns, clear ones to represent exons. Right? Your introns are the non-coding part of RNA. When we say non-coding, it simply means it doesn't code for a protein. They're just taking up space right now. Now, the point of this whole process is to increase efficiency. So if these introns do not code for the proteins, or do have a role in the mRNA, they're going to be cut out. Right? So the introns is going to be cut out, and you should just remain with exons. Right? So these are modifications made to the immature mRNA to make it mature mRNA. So this will be converted into a structure with only exons, right? Only containing the coding RNA. So now you get, so this is the immature mRNA. This will be the mature. Right? So these are modifications to the mRNA before it can leave the nucleus. Right? So just take down this diagram with the key. Right. Okay, so again, mRNA strand that is produced in your after transcription, directly after transcription, is called immature mRNA. This immature mRNA would contain both and introns. Right? The introns in your mRNA sequence doesn't code for any proteins. Right? So again, it's just taking up space. Right? In order to get mature RNA strands, you need to cut out these introns. Right? So what happens is these introns are cut out, right? Sent back to the, or just sent away, and you have mature mRNA, which only contains the exons. Right? So it's all you're doing as the modification is removing the introns. Right? Or the shaded part of the RNA. Right? So which strand do you expect to be longer? The immature strand or the mature strand? The immature strand. Right? Right? And this mature strand that would leave the nucleus. Right? Leave the nucleus. Right? Exons, introns. Hey, we're going on it. I was concerned, just I became distressed. Hmm? I mean, I spoke to his mom already. No, I didn't call him by name, but I think she knows just I was talking about. Right. So, okay, so just in the handout, if you're looking at the handout, right, do you want to send, I think it was last week or the week before, on the translation, you would see where they say, right, the introns are cut out, right, to produce this mature RNA. Right? Okay, so if you all remember the structure of the nucleus, if you remember the structure of the nucleus, you know to have ports, right? These mature mRNA molecules will leave through the ports, right? The mature mRNA molecule leaves through the pores and goes towards a ribosome, right, which is found in the cytoplasm. Right? Cheyenne, you understand the introns and exon parts, no?
Yes, sir.
Okay, and these introns are just removed by enzymes, right? You don't need to know the names of the enzymes, but the introns are just cut out by enzymes. Right. So after this, we could start with translation. I can erase this, right? Great. Now we can go to translation. So we transcribe our RNA. Now we can translate it. Okay, and it have, just like in transcription, it have three processes in translation. Anybody want to get those three steps? The same exact steps, right? Which makes it easy to remember. Right? So initiation. Huh? Yeah. Initiation, elongation, and then termination.
Maybe 6 hours, 6 hours. Right.
Initiation, elongation, termination. So remember, the mature mRNA left the nucleus now, right? It's in the cytoplasm. Okay, so it will go towards a ribosome. Secure, you want to draw it? Was a rhetorical question. Come on. Come on, the same thing. Are you drawing class? Yeah. You sure you could draw it, huh? All right. Right. So we'll go with this time, right? So this is this is now the tRNA actually looks, right? Remember, it have some more things on the side. For the simplicity's sake, we're going with this one. We are not number diagram. Look like a what? Can I spaceship? You can see. Anyway, right? Okay, so we in translation now, right? We have our mature mRNA strand, and it's going towards a ribosome. So remember, your ribosomes have two subunits, right? So that's what representing the two subunits here. Okay? This is our mRNA strand. And remember we said, well, I didn't say this before, sorry. Right? It starts at an AUG codon. Right? That's the initiation codon or the start codon. So what would happen is, at a ribosome, the mRNA is like passing through the ribosome. As it passes through the ribosome, tRNA molecules are going to be recruited. Now, each tRNA molecule has a specific sequence at the bottom. This sequence is called the anticodon. The reason it is called the anticodon is because it is opposite to the codon on the mRNA strand. Okay? So anticodon, mRNA strand. And if you look on the top here, you'll see an amino acid. This amino acid is methionine. This amino acid, okay, is formed from the anticodon. So based on the anticodon you have here, that will determine the aminos. Okay. Now, if you also check what AUG, what amino acid that produces. So just check in the table. Which amino acid does AUG produce? AUG. Remember, you'll have to check in the RNA one, right? Not the DNA one. Because the DNA one will have methionine, right? Right. So methionine, which is the start codon, we see AUG here, will code for methionine. So the amino acid attached to the tRNA must be complementary to the codon on the mRNA sequence. Okay? Yeah? Anybody confused? Okay. All we have, just let me understand what's going on here, right? We have our ribosome with cytoplasm. Okay. In the cytoplasm, we also have tRNA molecules. Each individual tRNA molecules have an amino acid attached to it, and it has an anticodon part attached to it. All of them are going to come together in the cytoplasm to form this complex, right, at the ribosome. So the mRNA is going to slide through the ribosome. As it's sliding through, you have these tRNA molecules that check it to see if it could pair up with any of the codons. These tRNA molecules come freely. So the first tRNA molecule that starts the sequence of protein production will be the one that contains the anticodon UAC. So this we need to start with this. So UAC will be looking for its complementary bases, which is AUG. Okay, that is how it knows where to start. So the tRNA finds its complementary pair or complementary codons, and it like stamps onto it. It sticks together. Each tRNA molecule also has an amino acid. Right? The amino acid is on the opposite side of the tRNA molecule. This amino acid is going to be complementary to the mRNA codon sequence. Okay, now we need to start with methionine. After this, let's just say we have UAC. Okay, what needs to happen is another tRNA needs to come in at this point, and it can't just be any tRNA. It has to be a tRNA with a codon complementary to this. So it's complementary to UAC. So it's complementary to U. So it's complementary to A and C. So this tRNA needs to have this anticodon. Okay? And how would we know what amino acid is attached to it? you need to find what amino acid UAC produces. So which amino acid is that? Right? Tyrosine, which is TY. Is THY or TY? TY. Okay? So the first step was initiation, right? The start codon, starting the entire process. The second step was elongation. So that's when the second tRNA came in with its complementary amino acid. Right? What's going to happen is this going to continue along the entire mRNA strand until it reaches a stop point. At that point, the amino acids going to form hydrogen bonds between each other and form your polypeptide. Okay? Then you have termination. You understand? These tRNA molecules are pre-made to match up with the mRNA. Okay? Right. So just take down this diagram. Well, you all have a note for it already, but I'll give you all the next one. The note from the handout. I don't know if you're looking confused from this, so something I should attention. Yeah. That's fine. Only was talking about micro. Only was talking about micro. Or you can see what I know. Oh, sorry. Forward. I'll tell a little. What the spike here? Why? Wait, if it's ever glass. It's logical. It's logical. I do the problems. Right, so after you finish the diagram, just a short note on translation, right? Again, it's on the handout. Not the same exact note, but it's saying the same thing. Yep. This one is have the peptidyl transferase. Yep.
Take us on a picture of the diagram, please.
What is the word next to central? Well, central dogma of molecular biology. So this word is dogma. Oh, you were saying something, Cheyenne?
Yes, sir, I was asking if you could send a picture of the diagram, please.
Oh, sure. Okay, so I'm just going to share something. Again, from the handouts. Right. So this is the initiation step, right? So you have initiation codon, which is AUG. right? So you have AUG. You have your ribosome. So what happens is when the ribosome realizes, okay, it have the initiation codon here or start codon, it's going to recruit a tRNA. Now we can see that this tRNA has the complementary base pairing of UAC. At the other end, you have methionine, which is the amino acid. So what's going to happen after that? The next sequence is CUG. So what amino acid is coded for by CUG? It'll be leucine. So another tRNA comes in and it have the leucine attached to it. What happens now is a peptide bond between methionine and leucine forms. Okay, and then the ribosome continues to move along the mRNA strand. Okay, it will continue moving. It brings along a third one, and we could see that every two tRNA, that passes, the other one going to be released. Right? So as one comes in, another one is released. But the amino acid stays because it's already formed the peptide bond. Okay? This process is going to keep repeating until we get this long string filled with a set of different amino acids. So this is going to continue until you reach a stop codon. In this case, it is UAC. At this point, the ribosome knows, okay, it has to stop. So it gets rid of this tRNA, and you're just left with the polypeptide chain. Okay, the polypeptide chain can then be used for whatever its role is, right? If it's an enzyme, it'll be used as an enzyme. If it's used to make up a type of hormone, it'll be used for that. Right, so this is a simplification of what happens. Right, you start off with nothing, and then we see the polypeptide chain slowly grows as you go along the mRNA strap. Right, the protein detaches from the ribosome, and then the process could repeat. Now, an mRNA could be used multiple times, right? It has a short lifespan, but it can be used to produce multiple polypeptides. Right? So the diagram wasn't as good as mine, but it's something, right? So you have it in your handout. They just take a note for it now. All right, so we're just taking a note for it. Okay, so again, the heading is still translation, right? Each tRNA, so right, each tRNA is attached. Each tRNA is attached. Each tRNA is attached to a specific. Each tRNA is attached to a specific amino acid. Attached to a specific amino acid based on its anticodon. Based on its anticodon. Full stop. Based on its anticodon. Full stop. During initiation, comma, the ribosomes during initiation, the ribosomes read the RNA strands. During initiation, the ribosome read the RNA strand until it finds until it finds the start codon of AUG. Till it finds the start codon of AUG. Let's stop. Right, let's stop. The anticodon pairs with the codon. The anticodon pairs with the codon on mRNA. The anticodon pairs with the codon on mRNA. And the process of elongation begins. And the process of elongation begins. Full stop. Right, just skip on a line. Peptide bonds form between amino acids. Peptide bond form between amino acids. To form a polypeptide chain. Peptide bonds form between amino acids to form a polypeptide chain. Full stop. This process is catalyzed. This process is catalyzed by the enzyme. This process is catalyzed by the enzyme peptidyl. Catalyzed by the enzyme peptidyl transferase. This process is catalyzed by the enzyme peptidyl transferase. Full stop. The tRNAs become detached. The tRNAs become detached from the amino acids. The tRNAs become detached from the amino acids. And leave the ribosome. Full stop. Termination occurs when the ribosome reaches a stop codon. which releases the polypeptide chain, which is the stop codon, which releases the polypeptide chain. Full stop. All the components are then disassembled. All the components are then disassembled. All the components are then disassembled. And translation is complete. And translation is complete. Full stop. Right, so just one more thing I want to go through before we go into the revision for module 1, right? The run through of module 1. Okay, so or we could just run, well, start, we're not going to finish it today, is something called mutations, right? Okay, so just something I want to introduce, right? This is mutations. Remember, a mutation is a change in the genetic sequence, right? It could be a random mutation, or it could be caused by a certain factor. But regardless, we're just going to go through different types of mutation, or introduce it at least. Right? So we could have two types of mutation. We could have chromosome mutations. Or we could have point mutations.
So like one mutation is a random change in the DNA.
It is. Any DNA sequence, right?
So you tell me I should have the same.
You just hit it on your arm, so.
So say you know everything about you and the first thing I'm saying answer was
You meant everything beside that one thing.
was up on but
No, just I can't help it. You on you. The one I'd given you was which one? What was the statement I'd given you on the class? It was he fought something. He fought no permutations. Remember, I gave all the original message and then he changed. Hmm? Yeah, yes. This was also he fought. Okay, right, so these are the type of mutations we're gonna have to look at, right? So point mutations are also known as gene mutations, right? So it could be gene or point mutations, and it could have chromosome mutations. So chromosome mutations is when you have a change in the chromosome number. Okay. So a change in the chromosome number will result in a disease like Down syndrome. Okay, you have an additional chromosome. Point mutations is when you're affecting the genes itself, the individual basis. Okay, so we'll go more in depth in the next class, but I just wanted to introduce it, right? So, like, so I said this is a genetic sequence, right? Yeah. Right, so let's say, remember, these are not actual bases, right? Right, because you don't have any bases with F, you don't have any bases with S, right? So we're just using it as a triple coda, as a message. Right, let's say this is the original message. We're going to use this as an example of point mutations or gene mutations. So let's say we have an insertion. Okay, so we have an insertion at the point insertion. Right, so we have an insertion at this point here. So we have fact remains the same, but instead of starting with the C, we have an insertion of S. Okay, so this becomes SCA. We inserted the S in front of the C. Now, what does this change? This changes the entire codon afterwards, right? Because remember, it read in triplets. The path will remain the same, but this will be read as SCA, right? So the other one would be TSA, and then TTH, and then EMA, and then you just have a T. It is the same message as above. So you all see how the insertion of one extra base could change the entire message. And you have to read it in triplets. Okay? So this is an example of an insertion of a base and how it could change or lead to a mutation. Okay? Another thing we could have is a deletion. Okay, so this one was inserted, right? Right? In deletion, what we're going to do, we delete C. So we're going to delete C, right? Somebody was asking. Yeah.
I wanted to find out if it's like if there's a reason that has happened or if that has just happened sometimes with no possible reason.
Remember, sometimes during DNA replication, the DNA is unzipped, right? So
Mhm.
what's happening is DNA polymerase is supposed to identify these errors and fix it one time. Okay, but sometimes, because the amount of things DNA polymerase have to do, it can miss it. Right? It's very rare, but it could happen. So the answer to your question is an error on the DNA polymerase part when the DNA unwinds.
Answer.
It can have different reasons too. For example, radiation, exposure to too much radiation, certain foods that you eat, right? It have a set of different reasons for causing these mutations. Right? Okay. So let's say we delete C, right? So we end up would the fat part be affected if we delete C? You know, because we're reading it in this direction, right? So be fat, but we delete the C this time. So we just have A, T, S, right? A, T, T, H, E, M, and then just A, T. So again, you change the entire message afterwards. Okay? Right, so that's insertion, deletion. You could also have substitution. And so which one we substitute in it? Right, so in substitution, but in substitution, only one of the letters is changed. Only one. So, right, so what we're going to substitute, instead of having C and cat, we're going to substitute the C with a P. So instead of having C, we substitute that with a P. We end up with fat, pat, sat, demat. Right, so this one might not be as bad, right? Because it doesn't change the rest of the genetic code. Okay, it might just change one amino acid or affect one of the codes. So remember that the genetic code is degenerate. So it have a possibility too that P18 might code for the same amino acid as C18. A possibility, but it's not likely. Right? Right? What else we have? Duplication. Okay, we'll go through the rest next class, right? Two more, but for now you can take a picture or write this down quick. Okay. Right. So we continue with mutations in next class, and then jump into cell division. You'll get a list of topics to focus on and just say module 1 picks up. Our exam was based on module 2 from module 1. From module, right? And we only did module 1. And no.
Yes.
We revised one topic in module 2. That was DNA replication. What was it? That didn't come in the exam. I told you all that wasn't coming. I told you all that wasn't coming because we didn't finish it then. Yeah, to include module 2. But they probably didn't reach that far as yet. You all start module 2 in school yet? Okay, well, their teacher taking it easy on them.
So we have 2,000 tomorrow.
Yeah, 2 labs due tomorrow and a test.
Okay, so what
But you don't have a test. Exactly. Where's our selection?
what level is in school combined?
We on lab 6 or 7? 6.
You have another, 12,
So we're done with labs.
but she's still.
Wait, so they do labs on topics so they didn't do yet?
She don't teach, because just go in class, do the lab, and she says study on model.
Yeah, I want to start to do that on
And she's going to get life with them.
Sure, it had a reason.
So you trying to skip class.
How much labs are they doing now? Unit 1, just the 12? How much are you all doing? 5? Wait, no, that's how much are they doing so far. But how much are they doing in total?
Still have that too, so you say some labs do stuff.
Right. I'm going to say that we're doing about, for bio, about 18. Nah, 12. Both of them is 12. But NAPS is do 18. All right, so we have it done. We could do the module 1 revision, right? Right. Right, so we're just gonna run through them, right? So we can cover as much topics as we could. I'm gonna share my screen. Share this to you. Right, so you could be using from these two documents, right?
So together study either from
Right.
people.
Oh, well, no, we it's just past papers are gonna use some of them. Okay, right, so we're just gonna run through the questions. If you know the answer, you could just say it, right? Right, so what are three structures, which this is, I believe this is the document, unit 1, midterm 1. Right, that is the first one that was sent. Second one that was sent. Right, so there's the second one. Right, so describe three structures which are common to a mitochondria and a chloroplast. So what three structures would they have in common? So think about the structure, remember, so it kind of have to do with the endosymbiont theory. Kind of related to it. So what three structures would they have in common? So mitochondria and chloroplast in particular. So remember, mitochondria and chloroplast had their own DNA, right? So remember with the endosymbiont theory, the theory was mitochondria and chloroplast was engulfed through endosymbiosis and it became part to form eukaryotes. So the first one, both of them have their own type of DNA. Okay, what type of DNA do they have again? Is it ATS or 7TS?
Right.
Okay, they would have 7TS DNA. So the first one, they'd have their own DNA. What's another one? So mitochondria and chloroplasts have their own DNA. Right? What else? They have their own membranes. So remember, they are membrane structures. Right? They have their own membranes. And what's one more thing? Own DNA have a membrane, both chloroplasts and mitochondria.
So I'm more for the
And this will be for on this after. Right? Anybody else? What are the structures they have in common? Online, you all have any idea? Seen any answers? Ribosomes, right? They have their own ribosome, they have their own protein producing machinery. So the answer for this one would be ribosome, DNA, specifically 7TS DNA, and their own cell membrane. Well, not cell membranes, they have their own membranes. Right? So part 2, how does a mitochondria differ from chloroplast with respect to function? So you just have to say the functions of the two. What's the main function of a mitochondria? To produce energy, right? So the mitochondria is responsible for producing energy, that is its function, or what about chloroplast? Its main function is to trap sunlight used in photosynthesis, right? Okay, so this one should be relatively simple. It's based on a food test, right? So just to review food tests. So you have some green and ripe green banana, half ripe banana, and ripe banana. You also have extract, well, water without banana. So they did the Fellings test, which is the same thing as the Benedict's test. Okay, and they also did the iodine test. So let's just observe the results, right? So for the iodine test, for the first green banana, we seen it have a blue-black color. What does that indicate? If you do an iodine test and you get a blue-black color, presence of starch, right? Right, and if it didn't have a blue-black color, if it had a brown color, it means starch is absent. So what observation we can make about, or inference we can make about green banana? Right, it has a lot of starch. Now you take a look at the same green or raw banana, right? What is the color change we get? Right, we have a greenish. So what does that mean? If a Benedict's or Fellings test have a red precipitate, starch is present, right? Okay, and you also have height of precipitate. So you have two results for that: height of precipitate and the color of the precipitate. Right, so they go on. In the half-ripe one, we basically get the same results for the Fellings test, except we have a bigger precipitate. So if you were to compare the reducing sugars in green and half-ripe banana, which one have more? Half-ripe, because it have a larger or bigger precipitate, a higher precipitate. Right? Ripe banana have the longest precipitate, and it have a pale blue-black color. So what that pale blue-black color indicates? Starch is present, but in higher amounts or low amounts? Low amounts. Okay. What's the point of using a banana of water without banana extract? What does it act as? Control, right? Would it be a positive or negative control? So remember, positive control would give you a positive result. A negative control would give you an expected negative result. So is this a negative or positive result? It'd be a negative result. So this would be a negative control. Okay. So let's get into the questions now. What is the hypothesis for this experiment? So remember, hypothesis, you're coming up with a statement that could be proven or disproven. What would be a good hypothesis for this? So it don't need to be correct. It just needs to be a statement based on the experiment. Right. Right. So which one you think would have the greatest level of starch? So we could half-ripe, okay. So half-ripe banana has the highest level of starch. Now we know from the results that's not the case, right? But yeah, the point of our hypothesis is to prove it or disprove it. So when you do the experiment, you can be like, okay, the hypothesis was wrong. This was the actual result. Okay? Right, so that's one possible one. Half-ripe banana had the highest level of starch. You could say ripe banana had the highest level of reducing sugars, anything along those lines. Once it could be proven or disproven. Okay, the next part, they want us to compare the results for the amount of reducing sugar and starch in the banana samples. So much reducing sugar. So this is 3 marks. So how would you compare them? So they wanted to compare the amount of reducing sugars and starch in each of the banana samples. So what would you state? Start by stating which has the highest reducing sugar content and which has the highest starch content, and then vice versa. Lowest reducing sugar content, lowest starch content. So which one has the highest starch content? Half-ripe. It would be the raw, the highest starch content. Remember, the iodine test is for starch, right? So the iodine test indicates that raw banana or green banana would have the highest starch content, right? What would have the highest reducing sugar content? Which one of these have the highest reducing sugar content? Right? It would be the ripe banana because of the color change as well as the height of the precipitate. Right? So ripe banana would have the highest reducing sugar content. And then you do the same thing for the lowest. So which one are the lowest starch content? The green are the highest starch content. So again, when you're doing starch, you're looking at the iodine test. A positive iodine test is a dark blue or black color. Right? So which one here had the lowest iodine content besides the one without banana extract? Which one? The right. Right? So the ripe banana would also have the lowest starch, and the green banana would have the lowest reducing sugar. And then the question asks you to state values, right? So you would state the values to support this. So for iodine, it will be the color change. For the reducing sugar, it will be the height of precipitate. Right, so what is the significance of the findings in this experiment in relation to bananas as a food item? So why is this useful? What would you use this information for? Why is it important to know, okay, what kind of nutrients the banana contains? What aspect of life is that useful? Diet, right? So if you have knowledge of the nutrients used or nutrients present, knowledge of nutrients present can be used to facilitate specific diets. So for example, right, if someone has diabetes, you would advise them to stay away from ripe bananas. Yeah? Because, well, reducing sugars that'll kill them. Right? If they want to eat bananas, you'd suggest they eat some green bananas. Okay, so you can use it in diet. What else could you use it in? Any other ideas? I think the main one here is diet. Well, what else could you use this information for? I think that's it in diets. Right, so use it in diets, right? For specific lifestyles. Right, and then part 4, we already answered this. What is the purpose of testing a sample with no extract? And that's so you have some type of control. So you know that all your reagents are working properly and you don't get any false positives or negatives. Okay, so the biochemistry part now. Identify the parts labeled 1 and 2 on amino acid B. So this 1, right? 1 would be what? So that is NH2. That is, what do you call that part? The amine or the amino group, right? So, and part 2. Right, so the acid or the carboxylic acid part. Right, so simple enough. Right, so they want us to draw a diagram in part 2 to show amino acids A and B form a dipeptide. Right, so when you're forming a dipeptide, at which parts do you form the dipeptide? The OH from the acid part and the H from which part? The other acid? The amine part. So I'm not going to draw it because of time. But what essentially you're going to have is the OH from the acid part and the H from the amino group. Right? What type of reaction is this? Condensation. So those two are going to be donated or given up to form water, and the amine group will be formed between the carbon and the nitrogen. Sorry. Yeah, ester linkage, right? Ester or peptide? Peptide, right? Peptide. Right, so we form a peptide linkage between the carbon and the nitrogen. So you're just drawing that back without the OH from amino acid A and the H from amino acid B. Right, so there's a good table here. Right, I want to hear from Josiah this time. Sorry, Joshua. Right, okay, so glucose, right? So the example they give us is amino acids, the linkage is peptides, and the polymer is protein. Right, so we are filling in the blanks. We have the monomer of glucose and the polymer is cellulose. So what type of linkages do you find in cellulose? So cellulose is the one we find in plants, right? So make sure it is. So what type of linkages we find in cellulose? I sent you all a table on this one. Think so. Alpha 1 to 4. Is that correct? What can we find in this? Beta 1 to 4, right? So in cellulose, we'd find beta 1 to 4. That is the conclude. Find beta 1 to 4. Where's my polysaccharides? Okay. So we'd find beta 1 to 4 glycosidic bonds. So B for beta 1 to 4 glycosidic. All right, so, okay. The next one, they give us amylose, right? So let's start with the monomer. What's the monomer for amylose? Alpha glucose. Yep. Alpha glucose. So A for alpha glucose. And what is the type of linkage? Right, so what type of 1 to 4? Alpha 1 to 4, right? That's it? Or what do you want to 6? No, right? So we'll have alpha 1 to 4. Again, is a glycosidic bond. Right, so you'll have to know these bonds. Right, in glycogen now, what will be the monomers? Or monomer? What's the monomer? Alpha glucose again, right? For glycogen. And what, so this time they tell us to have 2 types of linkages. So this should give it away. Alpha 1 to 6 glycosidic again, and alpha 1 to 4. Right, and alpha 1 to 4. So these are the type of linkages and the monomers that are used to make up the linkages. Right, we nearly done, nearly done. Right, part 3. So we could run through this. Outline the structure of a molecule of water and explain how it allows water to be an excellent solvent. So let's start with the structure. How would you describe the structure of water? It's made up of 1 hydrogen to, sorry, 2 hydrogens to 1 oxygen. The oxygen has what type of charge? Right, so the oxygen has a slightly negative charge and the hydrogen has a slightly positive charge. So that makes it a what? A dipole or polar substance. So because of this polarity, how does this help with its solvent properties? It's a solvent to what type of molecules? Well, polar molecules, right? So other polar molecules are molecules that carry a charge, for example, or slight charge. Example, sodium chloride. Okay, so that allows, so how does the water work? The oxygen does what? The oxygen attracts the positive ions, and the hydrogen attracts the negative ones. So that causes the molecule to split apart, sorry, the compound to split apart to be dissolved in water. Right? Explain all that to get your 5 marks. Now notice they only ask for solvent. They didn't ask about the freezing of water. They didn't ask about any of that. They specifically ask for solvent. Part B, what are 2 major rules of water in cell function? So remember, unless we went through the properties of water, just remember any 2 and explain it. So what does water help it in cell function? Right, so it acts as a transport medium, right? Because of its solvent property, it's an excellent transport medium. So that's 1. What else? Yeah, Chloe, you saying something? I was here and here to go just say, right? So it's the site of metabolic reactions. Remember, water has a neutral pH of 7. Because of that, it can be the site of many metabolic reactions. So that's 2 right there, right? Other ones would be the high specific heat capacity, which regulates the temperature in the cell. Doesn't allow it to get too high or too low. Okay? Right? So those are the 3 I would have gone for. No, any 2 out of the 3. And the last one, they want us to show the molecular structure of sucrose and show the position of oxygen and the other groups, except, so you basically don't have to give too much detail, just show the structure. So sucrose, right? What is sucrose made up of? Glucose and fructose, right? So you just have to draw the glucose and the fructose, right? Remember your oxygen up here, and you have your CH2OH up here. Again, your diagrams will be neater, right? And it's added to fructose. Now, you all don't need to do the before and after, right? I'm just doing it, well, because I'm teaching it or revising. Okay, right. So we have the glucose. That's the main part here. Once you show is glucose and fructose together, that's already a 2 marks. The other mark coming in for showing the correct type of linkage. So what is the linkage between glucose and fructose to form sucrose? What type of linkage? Right, so yeah, it'll be alpha 1 to 2. So the 1 from here, which is the carbon from here, would form a linkage with the second carbon here. Right, so 1 on the glucose would form a linkage with the second carbon there. Okay, 1 donates the OH, 1 donates the H, and you're just showing that linkage. Understand? Right. Wait, we're not done yet. Nearly. No, we have a next one. This is just one. I know. No, this is module 1. This is phospholipids. Right? We'll just run through it quick. So at least they'll have an idea what to revise. If they don't understand something.
So if you want, they could even.
Nah, they are to stay. Right? Right? Shakira, I know where to go. Ayana working on a drawing, and Chloe not leaving if all is still there. All right, we just running through it quick, right? So they want us to use a diagram to show the structure of a phospholipid. You don't, not chemical formula, just basic structure. What's the main things of a phospholipid, structure-wise? Right, so you have your phosphoric acid, or your phosphate group, attached to the glycerol backbone, right? And you have your 2 fatty acid tails. Okay, so they wanted the diagram that's about a 3 marks, and how you describe the structure. You would say it have a phosphoric acid head, which is which is hydrophilic or hydrophobic. The head is hydrophilic, and the tail is hydrophobic, right? So that's your 5 marks there. All right, so why are these lipids arranged in a bilayer?
Phosphoric acid and hydrophobic.
Sorry?
Hydrophobic.
The head, it's hydrophilic, right? The tail, hydrophobic, right? So the reason it's arranged in a phospholipid bilayer is because the hydrophilic heads needs to be on the outside, right? On the outside part is where it has the water, right? The hydrophobic tails, they interact with each other, right? Or they join up next to each other, right? They're trying to avoid the water. So the hydrophilic heads interact with the water or the outside part of the cell. The hydrophobic tails interact with each other, which is the non-polar part, right? So it have another tail here and another phosphoric acid down here. So this will be inside the bilayer, and outside of it, this part will be the cytoplasm, and this part would be the external part of the cell. So if you all see, the only thing that the tails interact with is other tails, because all of them are hydrophilic. Right. so just quickly, two ways in which endocytosis and I was joking, you all can leave it phone. All I want to, right? So two ways in which endocytosis and osmosis differ. So the first way is that endocytosis is an active process, and osmosis would be a passive process, meaning endocytosis require energy, and osmosis does not. Right? What's the next way in which they differ? Right? So osmosis is specifically used to transport water. Right? Endocytosis could be used to transport what? So you're bringing things inside the cell. You would use endocytosis for things like hormones, enzymes, any type of nutrients from outside the cell. So the two differences would be in structure, in function, right? And in one being active and one being passive. Right, so two examples of endocytosis for nutrients in animal system. So just say two nutrients that would be taken up by endocytosis. So what are two nutrients that endocytosis will take up? Huh? Hormones. And you can say glucose, right?
So if it's just hormones or the main.
You can just say hormones. The main thing they do, you cannot say things like sodium and magnesium because they are charged. Okay, they wouldn't enter through endocytosis. They'll enter through protein channels. Right? So quickly just running through. Right. So what are four main functions of proteins in living organisms? Right? So you have growth. We won't use energy. You use your sugars for that. Right? So you have growth, you have cell repair. What else? Those are the two main ones.
So they like muscle. Yeah. So
Hmm?
I say muscle.
Yeah, you can't say that. So because it's still repair, right?
Just give it like a general practices. Physical.
Yeah, so it does, but actually, yeah, we could put up, right? Gives organisms characteristics. What else? We need one more. Right, so remember, enzymes are types of proteins. So it's used to make enzymes. Remember, we also have proteins where? In our cell membrane, in the phospholipid membrane. So it's also used as transport medium in membrane. So that's the next one. Right? You also use proteins to make hormones. So you have a set of uses here, you just had to pick four. All right. So you have they give you the formula this time, right? For a polypeptide. So they want us to circle the polypeptide bond. Where would the polypeptide bond be here? So it have a couple, but where's one of the polypeptide bonds? So a polypeptide bond is between which two elements?
So that's.
Carbon and?
Mhm.
So the amino, right? So it'll be between carbon and nitrogen. Just remember, anytime you talk about peptide amino acids, proteins, it must have some type of nitrogen present. Okay? All right, so in the boxes below, draw the structure of the amino group and a carboxylic group. So that's from the last question, right? That would be here. Where you going, Mike? Right, so this is your amino group here, your NH2, and the carboxylic acid group will be a COH. So when they tell you to draw that, you just draw it out in the boxes here. Okay, so enzymes have many properties. State one properties of enzyme related to structure and one related to the main function. So what's a property of enzyme related to the structure? Yeah. right? So enzymes are substrate specific, right? So you just have to state it, right? You don't have to explain it for this one, right? So you just state it, enzymes are substrate specific. So that's related to the structure. And what about the function? One property of enzymes related to the function. They operate at optimum temperature ranges or optimum pH ranges. So that's related to their function. Okay, and another one, sorry. This one now, they want to sketch two curves, one with an enzyme and without an enzyme. So the one in red, the one in red and one in blue. So the one in red, is that with an enzyme or without an enzyme? Right, so the one in red is without enzyme, the one in blue is with enzyme, the one in red is without. Okay, the main thing here, remember it always requires less energy with enzymes. Okay, but the products are always the same. So you have to end at the same spot. Right, so this last one is just on an inhibition. Okay, so you have two graphs here, right? Reaction rate on the Y, substrate concentration on the X. So in the first graph, well, in both graphs with the inhibitor is the dashed line, without the inhibitor is the continuous line. Okay, so the first question, describe the trend seen in each graph. So in graph A, what is the trend? What is the trend here?
How many more?
Oh, okay. Just you can leave it here. Actually, no, next week now, before class. Right, so you're getting receipt and everything.
Yeah.
Right? Okay. Yeah, what's the trend in graphic? So you're just describing the shapes of the graph. So we compare with an inhibitor and without an inhibitor.
The substrate concentration will be less than the opposite.
So the substrate concentration increased, the reaction rate increased.
Yeah.
Right? Right? And compare that with the inhibitor now. So it's the same thing as reaction rating. So substrate increase, the reaction rate increase, but it increased at a slower rate. So you don't have to state why, you just state in the trend. Both cases, there was an increase, however, in the enzyme with an inhibitor, there was a slower rate of reaction up to a certain point. Up to which point that was? So up to which point did the graph kind of equalize? What value? Reaction rate 8 and substrate concentration of 60. Right? So you just stated that. Both had an increase, but with an inhibitor had a slower increase or slower reaction rate up to substrate concentration 60. Again, they just ask you to state the trend, so you don't have to give a reason as yet. Right, so in graph B now, how are we describing that trend? With the inhibitor, right? Was there any point of equalization where both of them had the same reaction rate? No, right? So you'd state that, right? With the inhibitor, enzyme with the inhibitor had a significantly slower reaction rate compared to without an inhibitor. And there was no point of equalization or no point at which both reaction rates were the same. Okay. So now they're going to ask you what the type of inhibition in both graphs. So remember, we have two types of inhibition. We'll tell you two types. Competitive and non-competitive. So you all would have to remember the characteristics of competitive inhibition. So let's start with graph B. Does this display competitive or non-competitive inhibition? So the key here is the substrate concentration part. Right, so this one will be competitive. Remember, with competitive inhibition, they're competing for the same sites. Right. And one of the characteristics of competitive inhibition was as substrate concentration increased, right, it would neutralize the effects of the inhibitor. Because yeah, if you increase in substrate concentration, it will reach a point where there's so much more substrates than inhibitors. So it will reach the active site first. So whenever you have a graph where there's a point of equalization, it means there's competitive inhibition. Substrate concentration increases, which decreases the effect of the inhibitor. So then graph P would be non-competitive inhibition. Remember, with non-competitive inhibition, right, they're not competing for the same active sites. So let's say the active site is here, right? The substrate's supposed to go in there, right? But what happens is there's another site down here where the inhibitor goes into. When the inhibitor goes into there, it changes the active site. That change in active site stops more substrate from entering. So no matter how much substrate concentration you increase, it's not going to change back the shape of the enzyme. So the rate of reaction would stay low. Right? Because the substrate can't fit into that active site. Right? So the only thing like these questions didn't cover really would have been microscopy. Right? So if you'll have to, well, you all should review that, right? I didn't have any questions here on the uses or types of microscope, which would be light and electron, the uses of them, and the differences between them. So, or could, well, it should be. Right? Is there anything with osmosis on this one? Yeah, well, then you'll have to go through movements as well, osmosis and diffusion, all that active transport. But those are just things to read with the water potential. Right? Okay, so that's it for